In this video, we will continue our discussion about this pancreatic cancer cells tumor microenvironment, and more importantly, this pancreatic ductal adenocarcinoma tumor cells. So, in the paper, they took the samples of the human patient who are not treated with this pancreatic tumor cells, and analyze those patients that are using cytoff. Cytoff is cytometer using time of flight. The advantage of this site-off is that they can use a large uh, panels. They can use a large markers in a single panel. That is around for example, they have used 40 markers in a single panels against the antibodies, and this is their method, which is represented in the panel A, where we can see that first they take the samples of the patients and react them with their panel of 40 markers, and then use this UV laser for this cytometer time of flight for the image acquisitions. And once they have image acquisitions, they did the cell segmentations based on the fluorescence, and and thereafter they did this phenotyping, that is uh, which cells belongs to which kind of categories, that is what kind of immune cells are this based on the markers. And after that they did their spatial analysis, that is they distinguished those cells based on the single cells RNA-seq data, based on the single cells data, that can be capable of this their technologies. Or what they did is, after phenotyping, they did the single cell data analysis to distinguish these cells into different categories. And after the single cell data analysis, they did the UMAP, so where they can see that the different kind of immune cells and different kind of uh, based on the different kind of markers are shown in this panel B. For example, NK cells are shown here, and CD4 plus cells and CD8 plus cells can be seen here, and the other categories are being identified. And they also did this heat map plot to find out in this kind of markers are present in different categories to which extent. That can be seen here, that is pan cytokeratin are more predominantly present in the epithelial cells, but not in the this alpha SMA plus fibroblast. Similarly, they did their heat map plots, and in the thereafter what they did is, so here they are able to see that each categories behave differently, or each class or segregated cells populations behave differently. And after that they also distinguish the cells based on the patients, that patients have what percentage of these immune cells. And the common things they are able to find out is that the NK cells populations are similar to the CD4 plus T cells or CD8 plus T cells, and this is true for most of the patients. And other things that they have identified is that epithelial cells and the fibroblast covers most populations of this pancreatic tumor cells, indicating that tumor microenvironment is mainly composed of this epithelial cells and fibroblast. Thereafter, the site of image is shown here of the whole experiment. Here we can see that the epithelial cells are present, and these fibroblasts are also present in predominant space, but the immune cells shown here are low. All the immune cells are shown here using yellow color, which are low. And this cytomap or the magnified image is shown here, which shows that which shows more clear image of that of this panel E, where we can see that even distinguish we can even distinguish this immune cells and the other different immune cells such as NK cells, and CD8 plus T cells and CD4 plus T cells can also be seen here in this cytomap. So this is how they do the spatial proteomics analysis of the tumor microenvironment of the pancreatic adenocarcinoma using CyTOF, that is cytometer time of flight uh, method. So I hope you understood in brief that what is the approach they used and what are the things, main conclusions that they find out. That is, NK cells populations are similar to the CD4 plus cells and CD8 plus cells combined, and most of the populations of the tumor microenvironment are composed of fibroblast and epithelial cells. So these are the main conclusions from this experiment. So I hope you understood this spatial transcriptomics uh, ma- uh, oh, experimental method and some of their analysis, some of the brief analysis results. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.